Good morning or good afternoon, everybody, and thank you for joining us for today's Care Connect session. My name is Lisa Saragusa. I'm going to take a few moments here to introduce who we are and what we're doing and what we're going to be talking about today. So again, welcome and thank you for joining us. If you want to take your camera off so we can see your smiling faces, that would be wonderful. But if not, completely understand. But our team at the care office, we have Myrna Gron, who is on the call today, but she is in transit, traveling from Washington back to Winnipeg today. Jordan Sisawa is on our call today. He is our marketing and industry relations manager. Diana Spenson, also on the call, is our office administrator coordinator. And as I mentioned, I'm Lisa Saragusa, and I take care of member relations as well as all our training needs at CARE. Tracy Shelton is not with us on the call today. She does all of our communications, so our new format of our digital newsletter. She takes care of putting that together for us, as well as our convention coming up in November. And then Juliana Gran is a part-time student who's helping us with our database and making that our CRM is up to date with everybody's information so that when we do have some critical information to send out to you, that it does get out to you in a timely and actually physically gets to you via email. If you are not on our database and you're not receiving our monthly newsletters, please reach out to any one of us in the office. Our contact information will be up shortly. And just let us know to add you to that list so that you don't miss information about these sessions or anything that's coming up that's industry related. On the screen, we also can see a list of all of our board members. So they too are happy to hear from you. If you have any questions about where CARE is going or what we are doing or what's happening with us, please feel free to reach out to a board member as well. So talking about contact and reaching out to us, here is our contact information. We are on most of the social channels, Facebook, LinkedIn, Instagram. We are YouTube, which is where all these recordings for the Care Connect sessions go. So please make sure that you follow us on all or one of those channels, like the post, share the post, comment on the post. Please engage with us. Phone number, email address, all that is there. And if you are on the call and you are not yet a member, we encourage you to help support us, help support the industry. So there's a QR code there. Use your camera to click that QR code, which will take you to the membership page where all the details are. And speaking about Care Connect session, which is why we are all here today. There's a series of Care Connect sessions, a lot of them being hosted and facilitated by AFC members, one of which is here today, but we also have some other great topics that are relevant for industry members. So if you do want to see our upcoming lineup, which is going up until the end of April, at which point we'll take a little bit of break because we know it's a busy season for our retailers, and then we'll come back again in the fall with a brand new lineup. So click that QR code to get to our upcoming sessions, and the ones that are available for registration, you just click on the link and to register. If it's not linkable yet, it will be shortly. But I do want to let you know, next week we have Christina Nick, I'm not quite sure how to pronounce her last name, but she's going to be talking to us about camellia agronomy. So if that's of interest, please join us for that. Now, let's get on with today's session. But before I introduce today's speaker, I do want to let you know that this is going to be an interactive, or we're hoping for it to be interactive. So if you do have a question, please put it in the chat box, and we will make sure that it gets addressed by our speaker today. But if you want to, once we get to the Q&A section, you raise your hand and we can call upon you and you can verbally ask your question as well. Again, we are here to ask questions and to learn about what the research is all about. And the person who is going to be telling us all about that is Dr. Crittenden, who has been a research scientist, thank you, in soil health and nutrient management at Agriculture and Agri-Food Canada in Brandon. Since 2017. His research aim is increasing agriculture productivity while ensuring the long-term environmental, sustainable, and agricultural systems through soil health and soil fertility management. Research currently focuses on assessing the utility of soil health indicators for Manitoba crops for our nutrient stewardship for P soil, physical quality, and proximal soil sensors. Stephen manages the Plant of Soil Nutrient Analysis Lab, sits on local and regional committees focused on soil management, is an associate editor at the Canadian Journal of Soil Science, and is adjunct professor at the Soil Science Department at the University of Manitoba. So that being said, I will hand it over to you, Stephen, and please carry on.
Great, thanks, Lisa. The first thing I realized looking at that picture you shared is that it's been a long time since we've seen summer sun in Manitoba, man. That nice canola was a long time ago.
We are looking forward to
it. Yeah, no, winter's really dragging on. So I'll just dive right in and share my screen, put it in presentation mode. And so I can't see you now, so you'll tell me if you can see it, the screen?
All is good, absolutely. Go right ahead.
Wonderful. Okay, so thanks for the invitation and thanks for everybody coming to the presentation today. I called the talk "Be Loyal to Your Soil," and I think if you sort of squint, it rhymes. Hopefully, hopefully that worked out. But I think the point is, that's one of the message I think that I'll try and give today is that thinking about giving importance to our soil is sort of the, if you want to say, foundation for our food systems, and I think will remain important. So you'd asked for some outline to the talk. So like any soil health talk, you have to give a definition of what it is, and then a couple of slides on sort of what's going on sort of politically, nationally with soil health, and a little bit in industry, and then a little bit more zooming into sort of soil health information from chemical labs. And then I'll run through maybe 15 slides or so, a couple of examples of some field experiments that I've been working in the last few years. And then I sort of pivot and talk about one specific soil health indicator, one property you can measure, and sort of tell the story of where it came from and for good or for bad, what it means or doesn't mean. So for me, soil health is about interactions of everything going on in soil. So soil is mineral particles with gaps between that has air and water, and when we measure what's going on there are biological properties, chemical properties, and physical properties, and sort of that interaction that is, for me, soil health. And we, rightly so, focus on our agronomic outcomes, our food production. But just one slide on to remind us the sort of broader context that managing our soils contribute to a sort of wide range of benefits, services, external to our farms, like climate regulation or water flood regulation, or even housing our cultural heritage, if you want to talk about archaeological sites. So just to sort of put soil health in the context, sort of broader context. But this is what I think about when I think about soil health, those interactions of things going on, those processes. So chemical processes like when phosphorus becomes plant available, desorbing, let's say, from minerals, soil minerals. Biological processes like the ones that regulate nitrogen mineralization from organic matter, and physical processes like the formation of soil structure and how water moves through soil. So that's what I mean. And a couple of sort of more in-depth, slightly in-depth examples. So here's a picture of an earthworm. I used to study earthworms, and they are really important for, they sort of burrow ingesting soil. They make large pores in the soil that contribute to water movement and aeration, but also to nutrient cycling, depending on the species. So this is one that we found, a student of mine found in Brandon a few years ago. I think when maybe more popular at the moment, when people think about the soil health, they think about the soil microbiome. So I'm not a microbiologist, I'll say that right now, but I have paired with Matt Backer, who's faculty at University of Manitoba, and this is some work that his student Mehrdad did at the site that I'll talk about in a moment. And it just sort of so the only thing I really wanted to say there is that from one year to the next, a dry year to a wet year, we were able to detect thousands more species. So I'll just comment that the soil microbiome is quite dynamic. Here's some physical properties that I measure in my lab. So how fast water moves into soil, how soil holds water. So if you want to talk about ability to avoid flooding or hold water during drought, we look at compaction and we look at aggregate stability. This is sort of how stable our soil is when you're trying to perturb it with water in this case. So all of those things are, so that's a benefit to resistance to change. And here's a picture. This is infiltration, basically. So the red lines are sort of a ring, and then we poured blue water, dyed water, into the soil. And then we made a soil profile. So that's what we're looking at. We're looking at the face of a profile, a soil pit. And what I find interesting here is sort of highlights the complexity of how water moves in soil. It doesn't just, move straight down. It sort of spiders in and out, following the lane of least resistance, if you will. And a lot of it goes down, but some of it goes to the side. So it's three-dimensional. So it's really just to say, and so this is where, like, if you imagine water moving through there and roots trying to find nutrients that are like nitrogen flowing through that water, that it is quite a complex system. So I think this is how most farmers interact with soil health information, like soil properties, chemical properties like nitrate and phosphorus and potassium. So how we decide on our rates of fertilizers. But as you'll see in a few slides, this information, the kind of information that's available on soil test reports, is evolving, and maybe evolving too quickly in some ways. Okay, so I wanted to just comment a couple of slides on sort of national picture about soil health and the sort of what's going on at that level. So if you're not aware, the Soil Conservation Council of Canada and the partners listed there are working on a national soil health strategy. Take a look at that online. So this is what they're doing. It's an industry-led framework for collective action around soil health at a national scale. So, like I said, every good soil health talk or project has to define what soil health is. So that's what they're going to do. Look at how we should be measuring, assessing at various scales soil health, and then, well, I presume looking for money, and then trying to sort of give advice and govern how soil is managed across the country. And on the same sort of note there, if you weren't aware, look this up online. There was a Senate report led by Senator Rob Black that came out last year called Critical Ground: Why Soil is Essential to Canada's Economic, Environmental, Human and Social Health. So they went across the country and interviewed soil experts at universities and governments and more across the country, got their opinion on what are the most important challenges to soil health. I believe the second reading of the bill was last fall. So I presume that there will be some updates on this in 2026. So watch out for that. Exciting potential for coordination on soil management nationally. And soil health also sort of pops up in ag media and industries interested. Here's a couple of quotes I just randomly pulled from the internet about soil being very complex and we have to try and understand it, and that soil health is a sort of starting point for yields, maximum yields. And the U.S. Department of Agriculture also chimes in on soil health, that saying that healthy soils are high performing and productive. They protect natural resources and they reduce production costs. So I guess I would say that that's a lot. That's already a lot, right? We haven't got to any of the science yet, or really how we interpret soil health information that is available to farmers. But so there's political interest, there's industry interest, media interest in soil health, and I think all of that is good. I think attention on soil is going to lead to better management, for better productivity for growers and better long-term sustainability. It's sort of it's more sort of that complexity and maybe inundation, if you want, that I think becomes a trouble. So or a challenge. This is an example I pulled from the internet of a soil test report that a farmer might get that has soil health properties, and then sort of gives you a score, a sort of overall indication of how you're doing. And I like this one just because it looks like a speedometer. So how do they come up with that? You sort of you send your soil away, or your agronomist does, to a lab, and then they measure a bunch of things, and they say, well, we think that more organic matter is better. So you sort of give that more is better kind of a function. Then certain things like salt, less salt is probably always better. And something like pH probably has a happy middle for where the majority of plants, majority of nutrients would be sort of most available, let's say. So that's a sort of valuation of what's on your soil test. So again, so to now that we saw that, we'll jump back to another example. This is an example from the Cornell University in New York State, Cornell Assessment of Soil Health, CASH, and they followed what I described earlier: physical, biological, and chemical properties. They sort of, you would send your sample off, as I said, you sort of get a measurement, and then based on the math behind the scenes, the valuing, it's either good or bad, and then you sort of sum up to get one number, or that's the idea anyway. But my impression is, and I think that's out there, that farmers don't know how to use that information to make management decisions. How does all that stuff you can pay for and measure in your soil test, how do you use it? What does it mean? And so that's what we have been trying to work towards the last few years, and that I will talk about in the sort of first half of the talk, about some ongoing work, some finished work and some ongoing work. Let's say trying to understand the utility. So how do you use what do they relate to? What are those soil properties you can measure? What do they relate to in terms of agronomic outcome, like crop yield, seed protein, and seed oil content? So here's an example from Saskatchewan. This is out of Kate Congre's lab at the University of Saskatchewan. This is an incredible work that they went, I believe, 50 fields or more over a 10-year period, and they measured a bunch of things. And they developed those curves I showed earlier were those are their functions. That's the math that they developed to assign a value to their measurement, their soil health measurements. And so what this table shows is that, in the strictest statistical sense, only 1 year of 10 did our soil health score, so our best scientific guess at how good your soil is, only one time out of 10 did that agree with crop yield, as in soil health was going up and yield was going up. And then if you want to be a little bit less strict, maybe 3 out of 10. But most times they don't agree. The soil health score with your crop yield in this case. And just to give another example, this is again cash, the Cornell system, and sweet corn here, and then corn yield. I just flipped it. Doesn't really matter. It's the same thing. Basically, just to say this is in North Carolina, I think it was, to say that the Cornell system score didn't relate to corn yield, and this is over 2 years. And so this is where we were trying to tease that apart. Those our best guess is the scoring functions that to the component parts, and trying to understand how that works, how those interactions work. So I'll show just a little bit of data from 2 field experiments in Manitoba, a tillage experiment at the AFC site in Portage, closer to Winnipeg, and then a short-term crop sequence experiment at the Ag Canada Centre at Morden. So firstly on the tillage systems, and so I don't know where the audience is. I mean, I think you're across the country, but so I think it's maybe relevant to remind, I think sometimes I hear that a sort of blanket statement about the prairies to say it's all no-till, but in fact, so this is a table from Cindy Grant and Don Flayton's work in 2019. It's Stats Canada data, and then I added that column, updated the data from the most recent Ag Census data to say that, in Saskatchewan and Alberta, 2/3 up to 3/4 of farmers report their tillage system to be no-till. But in Manitoba, it's only about 30% of fields. So that might be that heavy wet soils compared to not so heavy and wet. But I think, to me, this means that there's still a question, there's still a room for trying to understand your choice of tillage system and soil disturbance on soil health and crop productivity. And so that's what we did in this experiment that had run for 10 years. And these are just some pictures of the implements we used to create the sort of levels of disturbance. So a cultivation is one treatment, a deep ripping, a vertical tillage, and then a more disturbed system that I didn't put a picture there because it's sort of it's like at raised beds. We said at the bottom, it's like potatoes, basically. It's a subsoiling, a rototilling, and a shaping as if to look like raised beds or potatoes. And this is to get a sense of how representative the growing conditions were in those years. I really like these graphs. This is a water year, so it's from harvest, October, to next harvest, October. And this is cumulative precipitation, so just adding up the level of rainfall. And the black line is the 50-year median. So the colored lines just mean that in 2 of the 3 years that we were doing the study, the precip was at or drier than the 90th percentile, so really dry. And then 1 year was really wet, so at or sort of above the gray there on the top, so really wet. So that's farming, that's life, that's definitely research, dealing with variable conditions. Here's a list of what we measured. So and I'm not going to talk about all of them. I just thought I would list them in case, out of interest. So we picked commercially available soil health tests in my region because this is kind of the point, right, to try and help farmers understand what the soil tests mean or don't mean. And so conventional things like pH and nitrate and Olsen P, potassium, sulfur, and also some sort of newer, fancy, shinier soil tests like CO2. That's the Solvita burst test, soil respiration, and active carbon. That's the POXC, and ACE protein. Those are sort of some new ones. And we'll talk about one of those later on. In terms of crop yield numbers, that the tillage only influenced the yield in one of the 2 dry years. There's an interaction between what the crop is, the moisture, and the level of disturbance. This is a protein, just sort of out of interest, that the vertical tillage, so that's just where you run over the top of the soil really quickly in a very shallow system, tended to have lower protein, and I presume that's because of temperature for soybeans. And in terms of what those tillage systems influenced, well, soil nitrate was the thing that was influenced the most, sort of most frequently of all those things that we measured, probably because if you're changing the soil structure through your tillage because of the level of disturbance, then you're changing how water flows and where nitrogen moves. And then, of course, I think the vertical tillage there at the bottom was tended to be lower. Yeah, so this is a complicated slide, I know, but what I wanted to say is that in terms of sort of getting towards what of all those things that we measured, what related most to the agronomic outcomes was soil organic carbon, more than any other property. And so I'm not going to go through the sort of numbers there, but I'm going to focus in on soybean protein and oil content related to soil organic carbon. So these big arrows just mean that in 1 year, protein was sort of negatively related, as in like more protein, less carbon, or the other way around. And then in 1 year, the next year, it was the other way. It was more protein, more carbon. So how can that be in a property like soil organic carbon that changes really slowly with time in general, unless you're putting a bunch of manure or compost or something, tends to change really slowly from year to year. But so what does change, of course, are the growing conditions that we mentioned, those moisture, the water availability, and then influencing your crop productivity. So that's to say that when we want to try and sort of summarize, whether it's sort of into one simple number like a soil health score, when we try to bring all of that information together, and we base our value on what we scientifically know is generally better. The actual sort of relation and how we interpret it in terms of benefit on the farm isn't consistent from year to year. So I think that's a sort of challenge to be sorted out in future work. So there's some, I think I said sort of most of this. Actually, in this case, soybeans were sort of more responsive to, or soil health indicators were correlated more with agronomy in the soybeans than the other 2 crops in this case. So that work there is available in the Canadian Journal of Soil Science if you want to read some more of the science behind it. And I wanted to give a quick example of some work that we've also done at the Morden Research Center, AFC Research Center, south of Winnipeg, on short-term crop rotations. So probably this group, we don't need to say why we rotate crops to manage diseases and insects and nutrient availability, but crop rotation studies take a long time, and so they're expensive. So as in, if your crop rotation, let's say, has 5 crops in the rotation, you need at least 5 years to get every crop in the rotation, and probably you should have more than that. So then it would be 10 years, for example. So what we did, we sort of picked a trade-off there. So what we did was to try and get a lot of combinations of crop sequences in a short amount of time. So what we did was we grew alfalfa, canola, corn, soybeans, wheat, sort of in an east-west direction in 1 year, so 2021 in this case. And then we, in the next year, drove those same crops, but sort of randomized which ones where, and drove and seeded them in a north-south direction. So you sort of get this 25 by 25 cube of 5 by 5 cube of 25 combinations. So short crop sequence combinations. Those are the same years that we mentioned before. So very dry 1 year and very wet the next year. And in terms of what we would imagine would happen, this is a slide from Manitoba Agriculture. They have some great information on their website. So highlighting the crops that we have here. So wheat, canola, soybean, and corn. They didn't go alphabetical like I did in my list. Basically, what you see here is what I think we intuitively assume is that when you repeat a crop, probably the disease or pest pressure sort of builds up. So the relative yield where you repeat the crop would tend to be lower. But actually, in our case, that's not what we found. We found that where we repeated the crop, we had pretty good yields relative to other things. And we saw also a yield bump from where soybean was the crop before. We saw a yield bump for canola, and we saw a yield bump for wheat. I'm just sort of going to fly through this a little bit. This is protein. So this is, I should have said, the crop in the first year vertically, and then the crop in the second year, 2021, horizontally. So where corn was the previous crop, we saw protein in the following crop reduced, and I presume that is a nitrogen effect. But it's interesting that it's sort of legacy, that it sort of stays over to the next crop. And again, I know I stuck a lot of stuff in one slide, but all I want to say, so again, there's the two crops there, crop in 2021, crop in 2022. And what I wanted to say here is that where corn was the crop in 2021, soil organic matter measured after the following crop was reduced. Soil organic carbon in a couple of cases, and permanganate oxidizable carbon, or active carbon in another case. So that's I think that's not a predict. So this is a short study. It's not to predict that putting corn in rotation is going to reduce the soil organic matter. It's saying that the sort of I think what's going on here is the carbon to nitrogen ratios in those residues mean that it sort of helps decomposition, and so you would reach a new sort of equilibrium with your organic matter dynamics if you have corn in the rotation. Anyway, that's what I think that says, and that's my sort of explanation from what I can read, what others have found. Again, nitrogen, maybe not surprising, was the strongest related to crop yield, and pH and phosphorus and sulfur afterwards were also very important. And again, nitrogen, organic carbon, and some others were important for seed protein. So to sum up sort of that half, the first half of the talk, to say that the direction and magnitude of the interactions of soil health with crop agronomic properties is crop and weather dependent. So as in, I think this is what I said earlier, that if you have a soil property that's a relatively stable one, but your growing conditions are flip-flopping from year to year, that's the driving force. And then so when you want to try and sum things up, the picture becomes much more complicated. And so that's why some of those scores, soil health scores, might look, might not apply in every growing conditions. They might look a bit wonky. Okay, so that's the end of the first part. And now I wanted to just give a small discussion on one specific indicator. And it's going to seem like I'm picking on one thing, but it's just because I think there's a sort of a nice story here that we can learn from. So active carbon, this is the POXC, permanganate oxidizable carbon, that I mentioned a little while ago. It's meant to be, so it is a fraction of the total. It measures a fraction of the total carbon pool, all the carbon that would be in soil. And it was meant to be sort of active. So as in, like I said, soil organic carbon, the total amount changes really slowly from year to year. And so if if you're a researcher or if you're a farmer interested in trying to build your sort of resiliency of your soil system and you want to build carbon, well, it takes a long time. You're probably 10 years or something like that to find a total difference in the stock, the total amount of carbon there. So this is why this metric was developed, sort of the canary in the coal mine for change, the thing that you would measure a change first in this. And there are examples out there. For example, this where it worked. This example is from Missouri. They so in the Midwest Corn Belt, I think, of the U.S., about 450 sites. So a lot of sites. They measured corn grain, and they found that the active carbon was important for corn grain productivity. And so I think interestingly, they decided on a threshold that you got your maximum grain productivity where your active carbon, your POXC was 415 milligrams per kilogram. This is a chart of a histogram of our data from Manitoba. So these are our AFC sites, so with Oscar Molina, with our U of M sites with Yvonne Lawley in plant agriculture there. These are this is our median. So our middle is 800 ppm compared to 415 ppm. So where in Missouri they said you get maximum grain production from corn if it's 415. So what I want to say here is that taking a novel soil health test from one place and applying it to another place, especially in our case where I think our organic matter levels in Manitoba anyway are much higher, the total amount, than they are in Missouri, you can't necessarily translate those the tests or the interpretation from one place to another. So that's sort of one point on this active carbon. This is a chart from AgVise Laboratories there in North Dakota. They post their sort of summaries of the data. This is their distribution for the upper Midwest and Great Plains of the US. So it's ever so slightly slower, but pretty similar to what we found in Manitoba. So that's to say that I think our Manitoba data was pretty representative. So point two that I wanted to make about active carbon, these are the active carbon numbers related to our crop yield. This is in our tillage experiment, and it's flatlined. So it's there's no relation between crop yield and active carbon. It didn't matter what number the active carbon was, yield didn't change. And that's pretty similar. I know it's small, there's lots of detail there, it doesn't really matter. It's just there's a bunch of lines and most of them are flat. This is the crop sequence experiment in Morden. Although there were some statistically significant relations here, but again, some of them are positive and some of them are negative. So, but mostly no relation between that active carbon fraction and crop yield. So that's point two. Point three would be this is a study from 2023, sort of a lab study where they wanted to know what that chemistry, that active carbon method developed in 2003 by Ray Wheel's lab out of Maryland, I forgot to mention, what that method measures. So they this group here took several carbon compounds and they applied this method, and they said, well, actually, there are quite a few carbon compounds that the method measures or senses, let's say. One of them is lignin, for example, that is slowly degradable. I always think of it as the like in your salads or your celery, the bit that is really chewy. It's the stuff that decomposes really slowly. So in other words, it's the method that was supposed to be the canary in the coal mine that was supposed to tell us the active fraction to predict something later is not measuring that necessarily that fraction. So there's some uncertainty about what actually the method is measuring. And these are and actually the if we're So the active carbon is about a 1% of the soil organic matter fraction. So a very small amount. Another reason why if you have a big change in it, then you could probably detect it, whereas soil organic matter is much bigger and you need a big change to really sort of detect it. These are our data from across southern Manitoba, and again, sharing sites with Oscar Molina and Yvonne Lawley. But basically, probably there's some site-specific calibration, but more or less, you can say that there's a strong agreement between the soil organic matter and permanganate oxidizable carbon. So maybe you don't need to measure that sort of new fancy soil health indicator. And you look at the price, well, actually, it's about $31 per sample for the active carbon, the permanganate oxidizable carbon, and $6 or so. I mean, this is one example from one lab, but, you know, to give you a sense for soil organic matter. And in our case, soil organic matter was much more indicative of the agronomic response than was the active carbon. So it's okay. So I mean, I'm picking on one indicator here, and I'll give you a couple of justifications later about why you might still use it. But really, what I want to say is that before you start spending your precious dollars on any one specific fancy new soil test, that look at the backing of it and try to understand the value and the meaning and the interpretation. And just one slide before I sort of wrap up on how does soil health fit into this concept of Liebig's law of the minimum. I think the agronomists in the room will know about this. The idea is that what is the most limiting factor for crop productivity? So water in this case, and then nitrogen, potassium, sulfur. And I guess I would say that first, maybe it's important to emphasize that soil health, that all of those things are soil health, you know, sort of in my definition. And anything else you can sort of measure in soil can be mapped onto this kind of concept, you know, sort of with some calibration. So I think that's important to say, because maybe some of us might imagine that soil health is only the microbiome, or is only, again, those fancy new tests. And well, I guess what I'm saying is that nitrogen and pH and organic matter are still really important things to be measured in soil. So you know, even though I did pick on one test there in the second half, I think you can still find value in those soil tests, but you know, within your field, especially if you're going to make a management change, like a rotation change, or you're going to no-till, or you're going to start applying manure or compost or something, you know, take some soil samples and then come back after some time so you can track changes with time. And think about the variability within your field if you want to try and assess soil health. Think about the topography, think about salinity or erosion. So to try and get the best sort of estimation, you know, don't ignore those extremes. Maybe try and incorporate them. So as I said, yeah, soil tests don't guess. That's an old adage, and I think those oldies are still goodies in terms of specific soil properties that you should measure, like nitrogen, of course, phosphorus, potassium, soil texture, carbon, organic matter. And but I'll say really quickly. I think the conversation around soil health, I mean, what we talked about is sort of trying to help farmers interpret and understand the meaning and try to move forward with soil health information. But before you do that, you know, at least for me, it's sort of, I take it for a given that, but maybe it's important to emphasize, try and remove any sort of major restraints to soil health, as in your erosion. Is your soil eroding? Is it crusting? Do you have good drainage? Do you have salinity? You know, is there, well, any toxicity or pollution, or, you know, and then there's a whole range of things that aren't soil related, like weeds, disease, etc. Before you start, you know, trying to tweak your soil health, like make sure you are getting your those major constraints straight. Okay, and then to say being loyal to your soil, that one of my favorite authors says it all comes down to the soil. Get the soil right, and everything else follows. I think that's a bit of an exaggeration. Not everything else follows, but I mean, I think it is the basis for making good, sustainable agronomic decisions. And just to thank really quickly the technicians that worked on the project, and Curtis Cavers at Portage, Oscar Molina at Morden, and Yvonne Lawley at the University of Manitoba, and the funders, Manitoba government, Paulson soybean growers, and the corn growers in Manitoba. Great, thank you.
Thank you.
Oh, did I did I kick her out? Did she?
No, you know what? I will say this. We've had this once or twice in meetings where Lisa, we love her to pieces. We'll click the leave button instead of the unmute button. So
Did I?
No. This is a Lisa-ism. So what we'll do is I'll just take it until she jumps back. Oh,
I'm
there she
back.
is. Perfect.
Sorry about that. Yes, I actually hit the end button instead of the unmute button. Yes, sorry about that. Thank you, Stephen, first of all. But yes, first I guess what I'll do is I'll, again, thank you for that. Is there any questions from the audience? There were some questions that did come through the chat, but before I address those, I just thought I would open it up to those who are currently on the call. And again, if you're not too comfortable with unmuting your mic and asking your questions, my apologies, then go ahead and use the chat feature or the Q&A feature to ask your questions. And while we're waiting for that, I will pull up the questions that did come through the registration process, because I'm not seeing anything in the chat as of yet. So the first question that I was asked, the first participant was asking, I would appreciate any comments on soil microbiomes and how this directly influences nutrient density in food or crops, how these ties into human health, and livestock health as well.
That's a very broad question. I mean, it is a good one. So like I said, I'm not a microbiologist. So let me address sort of the soil half of the question maybe first, and I can comment really quickly on the others. I mean, so yeah, so like I said, the biological activity in the soil drives nutrient cycling, and it's so microbiology specifically. And what I saw, well, okay, let me say it another way. I had a microbiologist say to me once, and I very much hope you have one on the call that can refute what they say, but I had a microbiologist tell me once, like, oh, so actually you can just measure the nitrate test, or you can just measure soil organic matter. And this is sort of a broad way of saying that sort of the outcome, so those microbially driven cycles, if it's something related to nitrogen, the outcome is, from a plant's perspective, is the plant available nitrogen, so nitrate and ammonium. So if you sort of outcome, it would be sort of that before you get into a plant. And I guess it's sort of the same for sort of soil organic matter. So I know, so like I say, that's, I'm sure microbiologists would have sort of a different way of looking at that. And I guess I could, in terms of the, so human health is nutrient density in human health. This is quite maybe a jump too far for me, but I could point towards some work that we had published in the Canadian Journal of Plant Science a couple of years ago, which was about nitrogen, phosphorus, and sulfur fertilization. They were on canola fatty acid profiles. They were quite old varieties that took us a while to sort of get worked through and published. But what we saw there was that you could change fatty acid, the proportions of the components of the fatty acids to, you could change the compounds which were related to heart health, as an example. So if you want to say, can you draw a link? My interpretation is maybe not what they mean in this question, but I mean, so you can say our fertilization influences components of crops food that have an influence on human health. Maybe that's as close as I can get to.
And that's fair. Nope, that's absolutely fair. Thank you. And
Yep.
I still not seeing any questions, so I will ask the second question that came through the registration. And it said, should a producer be aware of certain products containing aluminum that may be harmful to their soil and crops?
Yeah. So, yeah, I don't work with heavy metals. And I guess my familiarity with aluminum is that there's quite, depending on where you are, there's quite a lot of aluminum inherently in soil relative to, say, metals like a zinc or something. Like, oh God, I'm gonna get the numbers wrong, but I mean it's hundreds times more aluminum than metals. I'm pretty sure. Like I think I looked up some numbers for you earlier, and it was maybe 300-400 part per million aluminum in, I think those are Manitoba soils. Was it that I looked up? But anyway, so there's a lot there anyway, and so the question is whether that translates to anything toxic. So I'm not really aware of uptake of aluminum per se. Certainly, if you talk about other metals, there's, I think, quite a can be a narrow range of before you get to toxicity of the plant. We did some work with zinc and barley, and in terms of well, in the context of making the barley better for fermentation and brewing and that kind of thing. That's not published. That's with Anna Badia, who is our barley breeder. But so yeah, we did see there that the level inherent in the soil of certain metals, nutrients, does change how much is in the seed. But I don't think I can comment on the toxicity for people necessarily.
Okay. No, and that's fair. Are there any other questions from the audience?
I got one that came in.
Okay.
Had an apology. Somebody had to leave in the middle. And basically the question boils down to if you have a farmer or producer that's unwilling to change their methodology on tillage, how do you best have a communication around soil testing and then moving more into the science of soil testing and supporting that way? I don't know if that, I don't know. I'm sorry if I'm butchering that question. Yeah.
Oh, well, I that's a sociology question. I mean, how do you, I don't know. I mean, I don't know how best to.
I guess the question would be more to how do you, if you have somebody that will not change the mechanics.
Yeah.
Okay, sorry.
Yeah, so no. I mean, twist their arm. No. I'm joking.
Jiu-jitsu.
Yeah. No, I mean, listen. So I guess I'm a scientist, and my role with the government is to provide evidence for decision making, influencing policy, and more importantly, how growers decide what they decide to do, how they decide to change their management. And so, I guess what I do when I interact directly with farmers, and we do some studies. For example, we've had questions. I had one farmer say, this was part of our Living Labs project. A farmer said, I have this, how'd they call it? A blob. They said, I have a nothing blob on my field. And they said, what's going on there? And so we did some measurements and we did some analyses, and then I gave a presentation just to those farmers to try and understand a little bit about what's going on there. So that was a case of someone who was interested in making a change and understanding why. But I guess my point is that trying to present the best information available so that they can make their own decisions. And I guess what maybe if I can extrapolate a bit farther after soil, I've worked now twice, like on specific research questions, with our economist, our ag economist, Mohammed Kakbazan. There's a publication, again, in the Canadian Journal of Plant Science on the economics of tillage systems in Manitoba. And we're also working with him on compaction on a soil compaction study. So a little bit less relevant from what I talked about today, but so in those, so what I'm saying is that we partner with, I mean, one thing is the soils question, another thing is the agronomy question, and the bottom line, I think, is the economics question. So that's why I try and work with the economists to say, you know, this is, we're piling on evidence about, that leads you in one direction. And so hopefully that can convince somebody.
Perfect. Yeah, and I think you're right. It's just the science-based or evidence-based to help sway a decision elsewhere. There is a few things in the chat. So one, thinking about all of these soil health indicators, it looks like there's plenty of data to look into, but I'm wondering about the coverage of potential gaps across the prairies, including microbiomes analysis. Are there efforts to provide a publicly available consolidated source of soil health data so that we can accurately compare against different areas?
This sounds like somebody from the Agri-Food Index group. So own up if it is, because I think they said they were going to ask something like this. So the fundamental question is that they're looking for aggregated efforts. I mean, I guess I would say this: I would be all for you know, collaborating and working together with people, researchers across the country. But I mean, I guess sort of the way, yeah, that sort of gets into how research works and how the funding works and how we try and answer specific questions. And we have had efforts within AFC, and there are efforts outside of AFC universities to try and put different data sources together to try and give a landscape, a better picture of what's going on with soil health sort of across the country. That's really difficult. Not that people wouldn't be willing to participate to share data. It's if you want to, one of the challenges there is that you need sort of a standardized methodology to do that. Otherwise, you could probably get data from different people across the country, but try and put it together, you might go, oh, actually, well, they measured this depth and they measured this depth, or they used this method that doesn't agree 100% with that other method. So it's a lot more difficult than what you say there. So you could do it two ways. You could just take all the information that could be donated, and then so those sources of variation, that error, take that as it comes, or you could try and plan to do something sort of standardized, which I think is the way that we're trying to go with some various other efforts.
Thank you. Oh, it was Chris Mancher. I'm not sure if you.
Canola. Yeah.
Who has that? This one's not necessarily a question, but there is a link in the chat from Janice, and it says this recent Newfield report might be relevant to questions about getting producers buy-in. So for that individual who asked that question.
Yeah, I will look at that as well. Thank you.
Yeah. But yeah, I don't see anything else. Anybody else have any questions? We are almost at the top of the hour, so made our full hour and had some wonderful information. Thank you, Stephen. So if there are no other questions, I will give everybody back their day. And again, I will thank you for joining us. Thank you for the conversation, Stephen. And I hope everybody has a great rest of the day.
Take care. Thanks very much.
Thank you.